Welcome to the University of Pittsburgh's Health and Explainable AI podcast. I'm your host, Jordan Gaspore, a health and science reporter. Join me as we cover advancements being made in health informatics and explainable AI for students, researchers, and healthcare practitioners interested in applications of artificial intelligence and machine learning. This podcast is produced by the University of Pittsburgh's Health and Explainable AI Research Laboratory at the University of Pittsburgh School of Health and Rehabilitation Sciences Department of Health Information Management. Headed by Ahmad Tafti, Pitt's HexAI Lab cultivates extramural collaborations with academic institutions both nationally and internationally through its research, educational contributions, and this podcast series.
Hello, and welcome back to Pitt HexAI, a podcast series produced by the University of Pittsburgh's Health and Explainable AI Research Laboratory. I'm Jamie Gramz, your host for today's session, with Dr. Tudorascu from the University of Pittsburgh, where we will discuss how neuroimaging statistics and AI are being used in research with the fight against neurodegenerative diseases like Alzheimer's. Dr. Tudorascu is a tenured associate professor within the Department of Psychiatry at the University of Pittsburgh and is recognized for her expertise in neuroimaging data analysis and imaging techniques. In her current and previous role as the associate professor of medicine at the University of Pittsburgh, she collaborates closely with colleagues in psychiatry, biostatistics, neurology, and radiology. She conducts research in three key areas, including improving brain tissue classifications in the presence of aging markers, imaging data harmonization, and reproducibility of neuroimaging studies for neurodegeneration and Alzheimer's disease. She serves as an expert advisor on study design and provides statistical expertise to multiple large studies, and is a co-director of the Biostatistics and Data Management Core of a large National Institute on Aging U19 grant looking at biomarkers of Alzheimer's disease in adults with Down syndrome. It is really great to have you on the Pitt HexAI podcast, and thank you for joining us.
Oh, thank you so much for the introduction, and thank you very much for the invitation. It's really an honor to talk about my work and my collaborative work, especially at the University of Pittsburgh.
Well, the honor and the pleasure is ours for sure. And maybe if you could just start off by telling us a little bit about yourself and your role as both a professor and a researcher at the University of Pittsburgh.
So I am a biostatistician by training. So my PhD was in biostatistics, and I got it here at the University of Pittsburgh, where I started my work while doing my PhD was in neuroimaging. That's what developed kind of like the trajectory of my career, specifically because I like how the statistics looks in brain imaging as opposed to just numbers. So I do a lot of work in specifically neuroimaging studies, but I also advise a lot of students in the ISP program as well as biostatistics. They work on my own research program as well as on the collaborative research that I have with my colleagues in psychiatry and neurology and several other institutions through big multi-site grant that I am co-directing the biostatistics core.
Really interesting. And given your experience collaborating with both researchers and clinicians across psychiatry, neurology, and radiology, can you explain why collaboration and research across these disciplines is so important for neurodegenerative disorders like Alzheimer's, and maybe share some examples of how it's being done?
So I think, first of all, I just classify myself as a collaborative statistician or applied biostatistician, specifically because I develop methods that are needed to solve real clinical problems. So, for example, when we first started working with brain data from people with Alzheimer's disease, and you look at these images, even if you're not a trained radiologist, but you can see specific differences between the MRIs of people that have Alzheimer's disease and people that have, just a normal looking brain. So in our line of research, most of the questions are arising from the fact what happens over time to these specific brains. So as a biostatistician, you have to quantify those changes using numbers. So the reason it's important to collaborate with people from different areas is that each one of us is an expert on a little piece. So unless we work together and we put everything together, there is no way we can solve these important clinical questions. So, for example, when I look at these images, I didn't know any anatomy of the brain before I started working with this brain, except very general things. But working closely with colleagues from radiology, I have actually learned how these changes occur, specifically with respect to anatomy. Working with psychiatrists, you understand what is the impact of the cognitive decline and the relationship with the neurodegeneration that is, that we can see in the actual changes that occur in the brain through different, imaging techniques such as MRI. And I work a lot with people at the PET Imaging Center here at the University of Pittsburgh, and we look at the functional imaging, which is specifically what happens chemically in the brain when you have certain molecules that are prone to, developing Alzheimer's disease, such as, amyloid plaque, and how is that image and how it's quantified and all those kind of things.
No, thank you. And with your expertise and background as a biostatistician, one of the ways that you collaborate includes training others in statistical methods and especially study design. Can you explain the role that study design and biostatistics play in clinical research?
Right. So first of all, we look at different things. So specifically, you're starting with a hypothesis. So when we have a clinical study, the most important thing in order for a biostatistician to develop an analysis plan is to work closely with a group of clinicians, neurologists, psychiatrists, and they are going to give you a certain question. I mean, based on that specific question, we elaborate in terms of how are we going to design that specific study. What kind of population are we targeting? What type of, what kind of age range we're interested in including in there. And specifically, it depends on the actual design. For example, in randomized controlled trials where you're testing the effect of an intervention such as medication or exercise, those studies are much more controlled than an observational study, where in an observational study, you just look at the general population and you observe, measure different things over time, and you see how that population is behaving over a certain period of time, or just one point in time, if you are just interested to look at some relationship between different markers of neurodegeneration and, cognitive measures or some other clinical variables that we are interested in. With respect to students, I would say that provides me a lot of joy, especially when they come and I see themselves, as I was a baby statistician, when they don't know where to start. And I think what makes me most happy is just to see that they are excited about the work. I think that's when they do really, really well. And it's very, it's comforting and it's very reassuring. And it's also a joy to see how much they enjoy answering questions. And when they write their first paper and then they struggle to publish it, or if it's not accepted, you can see how bummed they are. And then once they work through it, at the end of it, once they publish their first paper, they are obviously an expert on how to write it and give advice to the new students. I think it's really exciting, and I think it's the most rewarding part of my job to see them going through all these phases.
Well, it sounds like you're very passionate about the many students that you've helped throughout your career, and being an educator is a key area of what you do and the impact you've had. And your expertise is broader than just biostatistics. You also have broad medical imaging expertise. And part of your research within medical imaging, or using it, focuses on improving brain tissue classification in the presence of white matter lesions and atrophy in neuroimaging studies of people with age-related pathology or Alzheimer's disease. Can you share some insight on how brain tissue classification is actually done using medical imaging?
So, for example, when you look at the image that is coming from an MRI of a brain. As we get older, we develop these pathologies in the brain, which are called white matter hyperintensity. So there are different types of scans or different types of imaging techniques that we use. One of them is called T1 weighted, which is your regular MRI, and one of them is called T2 flare. So on a T2 flare, when you look at these, they appear as really bright, areas in the brain. And what happens is that when we classify the tissue, to do statistical analysis, we want to classify the brain as being, gray matter, white matter, or cerebrospinal fluid. What happens is that if you look at these white matter hyperintensity on a T2 flare, they're brighter, they're white. When you look at them on a T1 MRI, they are darker. So they look like gray matter. So when you segment, so what you're trying to do is you're trying to classify voxels in the actual image as being part of the gray matter, white matter, or CSF tissue. So these bright white spots, which are white matter hyperintensities, lesions in an older population, and they're very predominant in Alzheimer's disease and in female population, they appear darker in the MRI. So the computer doesn't know the difference, it just looks at the number and it says that since it appears to be the same number as the gray matter, it classifies it as a gray matter in the gray matter volume. But it's not actually gray matter, it's white matter. So over time, instead of correctly estimating this gray matter volume, it is artificially increased because we include white matter hyperintensity, because these are getting larger over time, but the gray matter tissue is getting atrophied, so you get less of that. However, we have developed techniques to be able to classify correctly, these white matter hyperintensities by using different types of, probability models or things like that. So I had several students working on this type of classification problem.
It's really impressive. And another area of your expertise also includes training others in statistical methods used for multi-modal imaging studies of neuropsychiatric disorders. Can you share some insight on what are multimodal imaging studies and why and how are they being used in research for disorders like Alzheimer's?
So, for example, we are not only using magnetic resonance imaging, we're also using PET imaging, which is positron emission tomography. So when people develop Alzheimer's disease, they have these, you have to excuse my ignorance because I'm not a clinician. So in general terms here, so they have these tag in their brain, which is called amyloid, or they have tau tangles in the brain. So we have different radio tracers that are actually specifically developed to target those areas in the brain. So when you inject an amyloid tracer, that tracer goes in, it's injected through your veins, it goes into your blood, and then we use positron emission tomography to image that so we can see where exactly it's depositing. So through, some specific quantification that our colleagues at the PET Center do, we can quantify that amount of amyloid in the brain through PET imaging. We can also quantify the amount of tau deposition in the brain through the PET imaging. And then we take that along with the magnetic resonance imaging and along with cognitive battery testing, and we can characterize and learn more about the disease. At different stages of the disease, we see the presence of these biomarkers. And now the statistics comes into play not only for, accurate quantification of these markers when you look at the longitudinally, or specifically when you try to figure out if it's in the right area of the brain or the images are co-registered specifically, very well together. But also when you develop, trajectory models. So you have to account for all sorts of factors, and you have to sometimes find different methods or develop new methods to be able to analyze this data accurately and estimate, the trajectory of the diseases or whatever other things that we're interested in estimating that arise specifically from clinical questions.
Thank you so much. And in multimodal imaging and even including data from other disciplines across diagnostic testing, you often hear about the need for data harmonization. Can you elaborate a little bit more on what data harmonization across modalities really means and share some of your experience on what's being done to make data from different disciplines available for use in research?
So data harmonization is one of my areas of expertise, actually. So that's what we've been doing for the past 7 years, specifically in MRI. So data harmonization refers to the fact that you have, let's say, in recent years, we are collecting massive amounts of data, and they're not collected from the same institution, right? They're collected from different institutions. So for example, we have a huge, study of Alzheimer's disease and Down syndrome. So we're collecting data from different institutions, meaning that different institutions, they have different MRI machines. MRI is not like a CT scan where you have standardized units of measurement. MRI has arbitrary units. So the numbers that you see at one institution for the subjects from a specific MRI machines are not going to be the same at a different institution from a different MRI machine, because the machines have different manufacturer, different Tesla power. So in that case, we have to put all these data together in order to make inference about the clinical population. So harmonization means that we have to combine data from different centers, from different studies, from different imaging MRIs in order to be able to make inference, to answer clinical questions. So in our case, we've worked specifically in MRI imaging. We use deep learning and statistical methods for data harmonization, but you can also develop methods of harmonization for PET imaging as well as diffusion tensor imaging. And data harmonization is not only applicable to imaging, it's also applicable to cognitive measures. It's also applicable to some other like blood biomarkers, proteomics, metabolomics, everything, because we use different machines to, separate plasma for the biomarkers and collect and measure that exactly, or even when you do cognitive measurements, right? Because you have different person that is doing the test with a different patient, we have to make sure that they are measured on the same scale and they are measured similarly, because otherwise the statistic is not going to be able to detect differences specific to the actual disease, but we're going to detect differences that are due to variability between the scanners or variability between the people that are measuring this, and not the actual difference in the measurement that is due to the clinical disease.
Thank you so much. And maybe if you don't mind elaborating a little bit more specifically for artificial intelligence and machine learning, how they're being used at some of your research to help with the diagnosis of Alzheimer's and other neurodegenerative diseases.
So I think that because we are working with high dimensional data sources that are coming from scanner, and not only from the scanner, as I said, from cognition, and specifically dealing with multi-scale studies that are coming from so many institutions, we have huge amount of data. So the deep learning methods are, what we use for these kind of studies. It is interesting because I learned a lot about the deep learning methods and these AI methodologies from my students, because as I said, I'm a trained biostatistician, so I learned with them. We used a lot for feature extractions. So if you have massive amount of data, specifically sometimes there is so much noise, and we found that the deep learning methods are specifically working well in classifying subjects with respect to certain features that we find in the brain. We also found that the deep learning methods were working very well for data harmonization. I recently had a student that published a paper on, deep learning methods for data harmonization. I think they have the capacity to work with these high dimensional images, and they are able to extract some features that we're not able to extract through specific statistical methods. They do not have the capacity to do that.
So thank you. And one of the other things I read was that you are the principal investigator of a National Institute on Aging funded grant focused on statistical methods to improve reproducibility and reduce technical variability in multimodal imaging studies of Alzheimer's disease. And that seems like it ties into the goal of the Pitt HexAI Laboratory, with the goal of trying to help make healthcare better through the power of explainable AI. So how does the research that you're doing in this area tie into explainability and trustworthy AI in healthcare?
So I think that a lot of the methods that we have developed, and especially the ones that I have written this grant for, were methods that stem from the clinical research that I was working with my collaborators. So I think it goes back to what we talked before, specifically with the data harmonization. I think one of the things that people are working and are trying to come up with are better treatments for these diseases, aggregating a lot of these data together, aggregating these multi-site studies and trying to extract information from it using deep learning or statistical methods, and even develop new methods and improved methods to answer these specific clinical questions. I think this is what is going to get us towards the finish line, such that we can seamlessly combine these data and we can make these methods reproducible. For example, I've seen before that you take a sample of subjects and you scan them on different machines, the same individuals, and you get different numbers. I think one thing that we're trying to do here is to eliminate those differences that are not differences that are due to the disease, but differences that are due specifically to technical variability, instruments that we're measuring the data with. I think in terms of the diagnosis and in terms of, how this tie into the specific healthcare, is that this is all contributing to developing medication, because this whole research is basically done to just figure out the best quantitative measurements that we extract from these data sets. So accurate measurements means that after we control all sources of variability, we can measure the effect of a treatment, the effect of medication. We can determine cutoffs for diagnosis groups with the help of our clinical colleagues.
You've achieved so much already in the impact that you've had, whether it's teaching your students or the research that you're doing, especially in the areas of neurodegenerative disease like Alzheimer's. Is there anything that you're really excited about in terms of projects, work you're doing, or future projects of where you hope to leave your impact as a researcher in this space?
I think I've always been excited about the work that I did. I think that to be successful, and especially to have sort of like to inspire students, you have to be really excited about what you do. I try to tell my children the same thing. If you're excited, you're going to like what you're doing, so you're going to be successful. I think one of the most important projects that I'm looking forward to is I have to write a renewal for my grant. I work on the first part on magnetic resonance imaging. I think the second part is going to be mostly on positron emission tomography. There are a lot of emerging radio tracer now that are measuring tau in the brain. So we have studies, there are multi-site studies with different PET tau tracers. So I'm actually looking forward to figure out how to combine these different studies with different tracers at different institutions. I have students that are now working with accelerometer data. So I think one area that I haven't done before was linking, the association, or looking at the association between these accelerometer devices and specifically, brain features in the brain that are extracted from different imaging techniques. I hope that most of our impact will be seen measuring treatment effects. I really hope that treatment of Alzheimer's disease is going to take off in the next 5 to 10 years due to the major strides that we've gone through, especially with imaging biomarkers as well as blood biomarkers. So I think we have the ability now to, through methods development and all these deep learning, to seamlessly integrate all these databases and learn more about future of the disease and be able to contribute in multiple ways.
Oh, thank you. And maybe reflecting back on your career and the journey that you've taken, are there any defining moments or experiences that helped shape the path that you've taken that maybe others that are trying to figure out what direction they want to go, that you would reflect upon and say that it had a defining moment or were key in your journey?
So I have to tell you that since I was a child, I wanted to be a medical doctor, like a physician. And then I did the undergraduate in mathematics. And I think when I came to the US, I was extremely excited to see the biostatistics program. That was actually what I think I was very passionate about, working in a clinical setting. And the fact that I had a math background, I thought that I will never be able to do that. However, the combination of the two, I think it's absolutely extraordinary. As I said, that was the moment where I knew that was exactly, the biostatistics program was exactly written for me. I think that sometimes I do see students that are not sure, in the beginning what it is that they want to do, or if they want to do something and then they are disappointed that did not work out the way they wanted. I think that in the first couple of years for a PhD student specifically, it's very hard to figure out what it is exactly that you do. But I think that, as I said before, there has to be a passion within yourself in order to be able to do a PhD and be successful, because that passion always reflects in the way you collaborate with people, in the way you teach others. And it also gives you that, you always want to learn more. And the more you learn, the more you figure out how little you know. That's kind of how it goes.
Well, it's very clear that you found your passion in life and you've been pursuing it and inspiring others along the way. And maybe regarding those lines, are there any key people that you'd like to maybe call out or recognize that were an inspiration to you or enabler of what you've been able to achieve throughout your career?
Yeah, absolutely. So I think one person that shaped my career, and especially my career in imaging, was Dr. Julie Price. She was my co-mentor along with Dr. Lisa Weisfeld. So Lisa was my PhD mentor, and then Dr. Price here at the University of Pittsburgh. Then I have a wonderful collaborator from which I've learned a lot at Johns Hopkins, which is Dr. Ciprian Crenicianu. And I think that I owe a lot to these three people. And I think one person that has been, in my opinion, is a legend in PET imaging. And I had the opportunity and honor to work with him. I've learned a lot from him. And especially, he's one of the most humble people that I have ever met in my life. And his name is Dr. Plunk. He is the developer of the Alzheimer Pittsburgh compound B that helped a lot in the Alzheimer research studies. That's why we have gone so far. So I think these are the people that have kind of made an impact. I think there are more, but I just can't remember now.
Oh, that's fantastic. And you're incredibly humble yourself with what you've achieved. But thanks for sharing some of the inspirations that have helped you along your way. And that, we'd like to conclude our discussion on today's podcast. And we hope that you've really enjoyed the discussion with Dr. Tudorascu. And Dr. Tudorascu, is there anything maybe you would like to close out? Any words of wisdom or guidance to any of our listeners, any of the students that might be listening before we end our session today?
Well, thank you so much. And it was very exciting to talk about this work. I think, especially for the students, I would just say to never be afraid to pursue their dreams. And every time they have an idea, even if that seems something that they will never be able to achieve, I think I would always try to think about it, even if you're not able to achieve the whole dream, but little by little, you never know where life is going to take you. I think there is also a lot of work in the neuroimaging for students that are interested in that. And one thing that I wanted to say is that in order to be successful with respect to this area and develop methods, I think you kind of have to learn how to work with images themselves and learn a little bit about the anatomy and about the disease when you try to develop methods for neuroimaging analysis.
Fantastic. So thanks again, Dr. Tudorascu, and thank you everyone for joining in today and listening to our interview with Dr. Tudorascu from the University of Pittsburgh.
Thanks everyone for tuning in and for following the show. The Health and Explainable AI podcast is produced by the University of Pittsburgh's Health and Explainable AI Research Laboratory at University of Pittsburgh School of Health and Rehabilitation Sciences Department of Health Information Management. I'm Jordan Gaspore. Thanks for listening.